Good afternoon, everyone. Thanks for coming to my presentation on Medicenna. Glad you're here. Medicenna, we are a publicly listed company on the TSX main board, also on the OTCQX. Symbol is MDNA or MDNAF. Just to let you know that I will be making forward-looking statements, so I encourage you to look at our regulatory filings when you have a chance. So what is Medicenna and what do we do? We are focused on developing life-changing immunotherapies for patients with late-stage diseases, particularly cancer that I'll talk about today, and other different immune-related diseases as well. So from a historical perspective, what we are really focused on is a class of molecules, we call them cytokines. So cytokines are immune messengers that we all have. And what we are trying to do is develop these cytokines that we have naturally, but generate what we call superkines. And the reason we do that is that we want our immune system to be best able to communicate with the immune cells and generate the best possible immune cells to fight cancer. That's the objective. And to do that, what we did was in 2016, we in-licensed this entire platform of superkines from scientists that had developed these superkines back at Stanford University. So we have exclusive worldwide rights to these very unique superkines from Stanford. We IPO'd on the main board of TSX in 2017. We have since then have a clinical collaboration with Merck, where Merck has agreed to provide us with a drug called Keytruda. It's the world's biggest selling drug. And that drug is being combined with our drug to see how these two work together. And then when we presented some early data last year at a conference, we had one of the world's largest oncology investors out of Boston, RA Capital, do a PIPE investment of $20 million. And what we're planning to do this year is, before the end of this year, provide you with an update on what we have done with one of these superkines, and this is MDNA 11. So we'll talk about that. And just to let you know that we have de-risked considerable amount of our development by virtue of two things. First, that we have agreement with the FDA on a phase 3 design for our brain cancer drug. I'll talk to you about that. And we have more than 100 patient data from patients that have received our drug with other types of tumors. So there's a lot of data that we have that we are leveraging. With Medicenna, we have an outstanding team of experts that have been working in the biotech space, in the immunology space. In fact, we have one of the world's #1 brain cancer specialists, John Sampson, as an advisor, been working with us for the past 10 years. Paulo Asieto, he's the #1 expert in Europe with skin cancer. He's been working with us for the past 5 years. And we have others who have decades of experience in running biotech companies, including myself. I've been at it for about 30 years. One of my companies had 1,000-bagger. So I'm really looking forward to really taking this particular program and getting some very substantial returns for our shareholders and of course, myself. So where are we with our pipeline? We have got a really exciting set of drugs that are in development, okay? And what we have right at the bottom is the drug for brain cancer. And this is a drug that is now ready for phase 3 clinical trials. And we're looking to partner this particular program. And I'll talk to you about the opportunity here with respect to MDNA 55, or we call it Bizaxofusp. We also have early stage programs, and I'll talk to you about one particular one, which is MDNA 113. And this is a drug which is a checkpoint inhibitor fused to an interleukin-2. And the reason I'm saying this to you today, although it's getting ready for the clinic, which should be in the clinic next year, is that this morning, Takeda did a $12 billion deal with a company called Innovent that is also developing a similar compound, but we think ours is better than what Innovent is developing. And then finally, today I'll talk to you about MDNA11, which is our IL-2 superagonist that is in the clinic. We have data from over 100 patients now. And as I said, a collaboration with Merck with this particular drug. And there again, this particular IL-2, the IL-2 space, has been part of multiple transactions worth multi-billion dollars over the past few years. So let's talk about what news flow we expect in the coming weeks and months. And the very top of the list is, of course, MDNA11. We have the bulk of data that we'll share with you all in December of this year. So towards the end of this year, presenting data at a major conference in the UK, where we will provide the up-to-date data on MDNA11 when it's used on its own or when it's used in combination with Merck's drug Keytruda. And also with MDNA113, which is the anti-PD-1 IL-2 fusion, which is a really exciting space, we'll have data from non-human primates as we get ready for entering the clinic next year with this product. And then finally, Bizaxofusp or MDNA55, this is a drug that we're looking to partner, and hopefully it'll bring in non-dilutive capital into the company. So let's start with MDNA11. This is as I say, the phase 1/2 program. We finished the phase 1 portion. We are now in phase 2 portion of the clinical trial. And what we have, just as a background, and tell you what IL-2 is all about. So IL-2 really is a interleukin that we all produce naturally in our bodies, and it is there to stimulate our immune cells to fight off infection, to fight off cancer, etc. And I can assure you, by the end of my presentation today, each one of you would have had at least one cancer in you. And the reason you won't have cancer is because these immune cells are fighting them off all the time. So we end up having cancer cells being generated regularly all the time in our bodies, but these immune cells fight them off. Unfortunately, when patients are sick or ill, the immune system is weak, it's not able to fight off cancer, and this is where IL-2 comes in place. So IL-2 was approved in about 20 years ago, drug called Proleukin, and that particular drug was used to treat patients with skin cancer and kidney cancer. Unfortunately, that particular drug, although it shrank and cured patients of cancer in about 5% of the patients, the problem was that there was an equally good chance that you could die of the drug because it was so toxic, okay? So the other thing was that the drug would clear from your bloodstream very, very quickly. So you'd have to treat the patient 3 times a day, every 8 hours for 5 days. And because it was so toxic, you had to be entered into an intensive care unit from the very beginning and get your treatment in an ICU. So a number of companies, Roche, you can see Bristol Myers, Sanofi, have all been engaged in the IL-2 space, trying to develop an IL-2 that's going to stay in your bloodstream much longer. Okay, so you don't have to treat patients 3 times a day. Hopefully you can treat patients once every 3 weeks, which they were successful in doing. But the problem was that you can see that they acquired these assets in these multi-billion-dollar transactions. The problem was that their IL-2s, although better designed, were not able to show tumor shrinkage. When you use their drug on their own, the tumors did not shrink. Okay, yes, they were safer. Yes, you could treat once every 3 weeks, but the tumors did not shrink, and therefore they abandoned this program. What we're doing at Medicenna is really addressing that big issue. Well, you can have a safer drug, but what's the point if it's not going to shrink your tumor or cure the tumor? And this is what we've been able to show in our studies so far, that with MDNA11, we are not only treating patients once every 2 or once every 3 weeks, but actually curing patients or shrinking tumors in a vast majority of patients. And these patients are not newly diagnosed. These patients have failed Keytruda. These have failed the big blockbuster drugs, and I'll share those data with you shortly. So if you look at the big market opportunity, where is it with MDNA 11? So if you look at on the left is Merck's Keytruda that's going off patent in 2028, in 3 years' time. Today is the world's biggest selling drug, nearly 30 billion in sales annually, higher and bigger sales than Ozempic. So this is a big drug, okay? And you can see that in patients that have previously failed therapies, only 16% of patients have tumor shrinkage. If you then look at Bristol Myers' drug, this is sort of taking two of their blockbuster drugs, combining them together, and even then, you have 20% of the patients responding to treatment. Whereas with MDNA 11, which has the IL-2 Superkine, fused to this albumin, we are seeing that we are able to shrink 30 to 50% of the patient's tumors, despite the fact these patients have failed checkpoint inhibitors. So what does MDNA 11 look like? So on the extreme left, what you see is the engineered IL-2 we brought in from Stanford. And what we've done is we've engineered it so that it's a lot more safer, and we've engineered it so that it's able to stimulate the cancer-fighting immune cells that we need. That's the key thing here. None of the others have ever done that. We've engineered it so that the binding of this molecule is dramatically improved. So the IL-2 that we have will bind to the cancer-fighting immune cells about 100 times more effectively than the IL-2 being developed by others. Okay, so that's the big thing. Another thing we did, which was very unique, was we fused our molecule to albumin so it can stay in the bloodstream much longer. So we made the molecule bigger, and this way we can treat patients once every 2 weeks or once every 3 weeks. But what is very interesting is albumin tends to accumulate in the tumor and in the lymph nodes that are close to the tumor. Okay? Now the lymph nodes is where you have your immune cells. That's where you train your immune cells. That's where you are able to generate immune cells that can recognize the tumor and attack it. And this is what ends up sort of creating what we think is a much better response with our drug than other IL-2s, or for that matter, other immunotherapies. So let me give you some examples here of what we've been able to show in tumors. So we've seen a number of patients. You can see tumor shrinkage, and these patients have all previously failed at least one, if not two, sometimes three different immunotherapies. These are end-stage patients. They have no other treatment options. These are the patients we treated. And we are seeing these kind of responses in these patients. And seeing that sort of response rate being so much better than checkpoint inhibitors is really exciting. And I think one important one that I need to bring up is the pancreatic cancer patient. So a pancreatic cancer patient had surgery. The patient then had a combination of 4 different chemotherapies together, and the tumor continued to grow in the liver. Stopped taking it, went to another combination of 2 different chemotherapies. The patient simply could not tolerate it. It was so toxic. Then the patient went to the mega blockbuster drug Keytruda from Merck. Again, that patient's tumor continued to grow in the liver, and this patient entered our clinical trial. Soon after receiving our first dose, these patients, we started to see the tumor stabilizing initially, starting to shrink, and eventually the tumor completely disappeared. These tumors were in the liver, which is the very difficult place to have a tumor shrink. But more importantly, this patient has stopped treatment with MDNA 11, or for that matter, any other drug, since December 2023. We are getting close to December 2025. There is no sign of the tumor coming back, which is really exciting. Have we cured this patient? I don't want to say we have, but it seems like we might have. We see the same thing with a cancer patient with melanoma. a complete cure of the tumor, despite the fact the patient failed multiple chemotherapies and Keytruda. So we're seeing a durable tumor shrinkage, which is not only shrinking and then growing back again in 6 weeks later. These tumors get shrink and they stay shrunk for months and months to end. And we're also seeing a very nice safety profile. We're not seeing the need for the patients to be in intensive care. These patients come and get treatment once every 2 or 3 weeks, and the majority of them receive their treatment pretty much on an outpatient basis. Okay. So where are we taking our drug? So you can see pancreatic cancer patients. That's really exciting. We are also seeing melanoma is an exciting space for us. But when we combine our drug with Keytruda, surprisingly, we saw that these patients had failed, Keytruda, and these are patients with endometrial cancer. This is really aggressive at this point, and we saw half of the patients respond to this drug as well. So we have multiple shots on goal with this particular drug, either on its own or in combination with other therapies. But let me give you an example of a sort of a benchmark as a comparator. Where are we from a valuation perspective? You can see that's where we are valuation-wise, about 60 million US, 80 million Canadian, and we are seeing response rates of 30% already with our drug on its own in patients who have failed checkpoint inhibitors. Look at these two other companies, Replimune and Iovance. Both of them have valuations that are 10 times or more higher than ours, seeing similar response rates. And these are difficult treatment procedures. With Iovance, you have to first extract the tumor from the patient. You have to isolate all the immune cells, grow the immune cells outside the tumor, outside the patient, inject it back in the patient. The patient has to be in hospital for 30 days, and there is a good chance, 1 in 15 chance, that the patient will die because of the treatment, okay? And they only get response rates of 30%, and it costs per patient $550,000. You got a 1 in 3 chance of having a response after spending more than $500,000. Replimune, on the other hand, has to have a combination of an oncolytic virus and a checkpoint inhibitor together. And on its own, FDA recently rejected their application for approval because they did not see their drug, when used on its own, was not able to shrink tumors. We, on the other hand, are seeing our drug cause tumor shrinkage in 30% of the patients with melanoma. So there is a huge opportunity for us right from the get-go. We are looking at, in our phase 2 trial that's currently underway, patients with melanoma, which is 7,000 to 10,000 patients available every year. And despite the small patient population, you can see sort of the $800 million valuation. MSI-high tumors, it's a 3 times bigger opportunity, and that's where we're seeing tumor responses as well. And then finally, another biomarker, which is TMB high, where the opportunity is about 10 times bigger than melanoma. So just think about it. We have valuation of about $800 million for these kind of programs, whereas we have 3 shots on goal, perhaps even endometrial cancer, I haven't included it here, potentially 4 shots on goal with data that's going to start coming out at the end of this year. So we will be positioning ourselves for multiple opportunities with this particular drug, and I believe this has a multi-billion-dollar opportunity for our shareholders and obviously benefit for our patients. Let's talk about something else. This is brand new, happened this morning, 113, okay? It is, as I said, a combination of anti-PD-1 and IL-2. It's a bispecific molecule. We call these BiSKITs, okay? So just to give you an idea, what's happening in this bispecific field, you can see these anti-PD-1s, multiple transactions occurring with this checkpoint inhibitor space. You can see these are multi-billion-dollar annual sales with a number of patents expiring, but look at right at the bottom here. You can see these bispecifics. These are just over the past 12 months. You've seen these multi-billion-dollar transactions taking place, but even more important is the transaction that occurred today. It's an anti-PD-1 fused to an IL-2. This is a program that was announced this morning between a Chinese company called Innovent and Takeda. The total deal size was over $11 billion with about $1.2 billion upfront. A $1.2 billion upfront. Now, if I look at the data from their work that they presented, they've shown a response rate in melanoma with their massive anti-PD-1 IL-2 drug of about 20 to 30%. We are seeing a 30% response rate with only the IL-2 portion of our drug, okay? So just imagine what we will be able to do with 113, where we have fused the IL-2 to our anti-checkpoint inhibitor. Now, the checkpoint inhibitor we are using is not a new checkpoint inhibitor. We're using the same checkpoint inhibitor that's going off patent. It's Keytruda and Opdivo. We are evaluating both, and we're going to test them in monkeys. The study is going on right now. We'll have data before the end of this year as to which will be the lead program. Is it going to be Keytruda or is it going to be Opdivo for the clinical trial? What we've done is we've taken the blockbuster checkpoint inhibitor, fused it to the best-in-class IL-2, that is the same IL-2 that you see in MDNA 11, and we have masked it with a drug called IL-13. This is an inactive drug, but it helps us to direct and localize the drug right at the tumor site. So when the drug is circulating in the bloodstream, this particular drug is inactive. It's not active because we are masking this molecule, okay? When it gets to the tumor, these pink squares come off. It's like a switch, comes off, and suddenly the drug becomes active, binds to the cancer-fighting immune cells, and the immune cells are now supercharged to attack the cancer. That's what we do with this particular drug. And the reason we use IL-13 is that it localizes the drug to the tumor because about 2 million cancer patients annually have tumors that have this particular target. And this target will only bind to this IL-13. So this is where we have the exciting data that we will be sharing at the end of the year and also next year as well. Finally, last but not least, is our brain tumor program. This is really a drug that has shown some really impressive results. Just to give you an idea from a background, 20 years ago, this drug was approved, Merck's drug, Temodar, and all it did was improve survival of brain cancer patients by 10 weeks and became a billion-dollar blockbuster for Merck. And this particular drug is even today used as the first-line treatment. It is the only treatment available. We, on the other hand, with our drug, have shown that we can actually not improve survival by 10 weeks, but we're improving survival by 7 or 8 months. Not in newly diagnosed patients, but patients who have failed surgery, failed radiation, failed Temodar, and got in our study, and we're extending the lives of these patients by 8 months. That is a big deal. And the opportunity is massive for this particular drug. You can see that even for the first line or second line or third line treatments, it's a $800 million opportunity. As you expand into different types of brain cancers, you can see the market opportunity is about $4 billion. Just from context, just to let you know, brain cancer or glioblastoma is uniformly fatal. We've heard about Gord Downie, the lead singer from Tragically Hip, who succumbed to this particular disease. We think we've got potentially the best drug for brain cancer as well. So multiple data readouts coming up this year, particularly before the end of this year. The company has cash runway into Q3 of next year. So we are looking to share more data. We've got good coverage now. We continue to build analyst coverage in the US as well. And importantly, to let you know that, as I said, RA Capital is the largest institutional shareholder in Medicenna. Insiders own 23% of Medicenna, so there is skin in the game from management. And as I said, multiple readouts coming up. All of these, the first 4 of them are going to be disclosed before the end of this year. So coming up in December at a conference happening in the UK, a major major cancer conference between 10th and 12th of December. So I'll leave it there and happy to answer any questions. Okay. Right. Yes.
You just finished phase 1, usually, order patients and was offensive power.
No, the studies were not powered. So these power studies will be part of a phase 2. So we have generated proof of concept now. So the next thing, so that's pretty much done now. We'll be ending by proof of concept by the end of this year. We are then planning to meet with the regulators, the FDA, to see if there's a pathway for a registration phase 2 trial. So the phase 2 trial will be a registration trial seeking accelerated approval. So it would be an adaptive design with 2 arms, or it could be just a single arm, because the primary endpoint for accelerated approval is tumor response, is tumor shrinkage. That's what we're looking for. No, ORR. That's what we're looking for. Yes. Yes, that's first line. The FDA will not. This is end-stage brain cancer patients, melanoma patients. So if you look at Replimune and Iovance, both of them got, or at least Iovance got approval based on 70 patient data in a single arm phase 2 clinical trial. That's it. So we're using the same approach. Because Iovance got approval based on a single phase 2 study, single arm phase 2 study, we are using the same approach. The difference being our drug is safer. It's obviously not going to cost $500,000, and it doesn't kill 1 in 15 patients, which is what Iovance's drug does. Right? Yes.
They said you use Keytruda doesn't work on about 1/3 of patients. Is that correct?
Well, Keytruda doesn't work in about 2/3 of the patients. So it works on 1/3 of the patients, and that's despite that you're seeing it's a $30 billion annual sales, and 2/3 of the patients don't respond, right?
Haven't checked for 2 years.
It will, yeah, the exact copy of Keytruda or Opdivo. So it'll do the same thing. So when they come out in 2028, they'll have the same results.
It would make sense that they would be your partner and understanding their life. You get how much money do you take this stuff away?
Absolutely, and others as well, potentially. Yeah. Yeah.
And just on the definition of response rate, 30%. If you look at the 11 of the 17 patients, it means 2/3 of patients either were flatlined, there was no change, but not a deterioration.
Yeah.
Improved and actually got a measure. Was that a 60% definition of response rate? And I'm trying to figure out whether is a response rate, or you actually have an improvement.
Yeah. No, so we set a certain very, very strict guideline. Tumor has to shrink by 30% or more. Okay, but yeah, so even if patients' tumors shrink by 25%, we don't call it tumor response, right? So it's that threshold. That's why when I say, yeah, you're right. When you looked at the graph I showed you, you saw that about 60% of the patients, either the tumor stopped growing or the tumor shrank by less than 30%. So that we did not include in our numbers. So if you include them, then it's about 60% clinical benefit rate. They call it clinical benefit. Okay.
Back in the experience of our IL-2 has better binding to the tumor by design. So that means it actually works better when it sticks like up to a blanket
Yeah,
And it basically attacks.
correct. It binds to the immune cells, not the tumor cells. The IL-2 binds to the immune cells, and the immune cells, it's basically essentially stimulating the immune cells to grow and be more efficient and faster and more ready to attack the tumor. In fact, what we see in some cases, we see that these immune cells, at least in animal studies, where we are able to inject the tumor again in the mouse, the same tumor will never show up, even after a year. When we inject the tumor again, because the immune cells now have memory, and they remember, just like a vaccine, that there is a tumor that has entered my body, it will attack it right away, and the tumor will never show up. So that's what we call really exciting about this approach. That's why we're seeing durability. That's why we're seeing patients, for instance, the pancreatic cancer patients, 2 years, no drug, no treatment, no sign of tumor. Because the immune cells, the memory immune cells, are there all the time, ready to attack as soon as the tumor comes out.
Instead of gets COVID, they can contact the Senate score.
Correct.
So too much of a thing.
Yes.
I would have said that normally operate the body kind of recurrent like
Yeah, so, the thing is, obviously, we wouldn't be using our drug for, say, COVID, for instance, but I think the key thing here is for certain infectious diseases where you have the opportunity, for instance, hepatitis is a good example. There we've seen evidence that an IL-2 kind of drug can also stimulate your immune system in such a way that it's going to attack those pathogens. Our focus is right now not on infectious disease or anything like that. We're focusing right now in cancer. Okay. Yes. Sorry? Bizaxofusp, we first started in 2021-22. We had an end of phase 2 meeting with the FDA. They showed us a pathway to get approval for this particular drug. But unfortunately, at about the same time, we had multiple failures in glioblastoma, including big pharma like Opdivo, Keytruda, et cetera, failing. And there was a I would say, over the past 30 years, there has been a carnage of failures out there. But when our data came out, we had these very big clinical trials, hundreds of patients in the clinical trials that failed. There was therefore reluctance for pharma to look at partnering. Things have changed this year, though. They're seeing, there's interest again in the brain cancer space, and we feel that there's an opportunity for us to take advantage of that. Okay. We have new data, but that's currently not published. It's being prepared together for a publication, and we'll be using the data to submit to the FDA as well. Okay. Yeah. So the next value inflection point for us is the data in December. December data is the next value inflection points. We have multiple data readouts coming up, first in sort of melanoma and these other tumor types, and then potentially other readouts as well. But these are the key readout coming up in the next 3 months. Yeah. So our burn rate is about 4 to 5 million Canadian. So we've got enough to take us into Q3 next year. This happened. So this was a deal between Takeda and a company called Innovent. Innovent is a company based in China. They have they've got a huge pipeline of molecules, but one of them that is generating a lot of excitement is the anti-PD-1 IL-2. That's the drug that they licensed or partnered with Takeda. So it's 1.2 billion upfront, and there's milestone payments, et cetera, that come in. That's up to another 10 billion. So makes it about 11.2 total. Okay. All right. Thank you very much. I appreciate that.